So this story starts about 6 years ago at The Ohio State University when our cancer doctors came to me and they said, we have a problem in breast cancer. And how many of you have heard of personalized medicine? Anybody? Okay. So this problem has to do with personalized medicine. And the problem is there's not one type of cancer. Well, we know that there's breast, there's lung, there's colorectal. But even within one subtype of cancer, like breast cancer, as you can see from this image, there are many different types of even breast cancer. And to identify which type a patient has, you have to look for these things called biomarkers, which are listed on the right, and there's a lot of them. And so we want to be able to look at all these different markers and determine which markers you have and which markers you have and you have, and then we want to tailor the treatment based on the different markers that you express. And so here's the problem. This is what we do right now. If a doctor thinks you have cancer, they will take a biopsy sample, and they'll take that sample and they section it and they put a piece of tissue on a slide and they stain it with something that's pink and purple, and it stains tissue and blood vessels, and they look at an image like this and say, the shape is irregular, you have cancer. Okay? The next step of that, to determine what type, would be to look for one of these biomarkers. And in order to do that, they use a brown stain, and wherever the brown stain appears, that's where the biomarker is. Now, the problem with this is I can only look at one marker at a time. And I may need to look at anywhere between 10 to 15 markers to really know what type of cancer you have and what type of treatment you should receive. So the way they do this right now is they take sequential slides. They take a brown image in each slide for a different marker, and then they overlay the images. And so they asked me, was there any way that I could look at all 10 or 15 markers all at once? So now we have to go back even further to when I started my PhD research in 1999. And I was studying a type of material known as quantum dots. And quantum dots are nanoparticles that are made from semiconductor materials like what you would find in your laptop. And they consist of somewhere between hundreds to thousands of atoms, unlike a molecule, which has maybe tens of atoms. And because of that, they have some unique properties. For example, they fluoresce. And what that means is when I shine light on them, a different color of light comes out. And I can make lots of different colors. So I can have red and green. And the color of the light is dependent on the size of the particle. So bigger particles are more red, and smaller particles are blue. That means it's really easy for me to make 12 to 15 colors, like I would need in this application, so that each marker could be a different color. Okay. In addition, they're crystalline materials, and their surfaces can be modified with molecules that will bind to these cancer biomarkers. So those molecules might be peptides, it could be a protein or an antibody, or it could be a piece of DNA. And it's fairly straightforward to put those molecules on the surface of these particles. So you would choose an appropriate molecule that would then identify the marker that you're looking for. So this is an example where we've taken a green/yellow quantum dot and we've bound it to a marker that binds nerve cells. And so the marker is on the outside of the nerve cell, and you can see everywhere where there's green or yellow, we have found that marker and we can identify that cell as a nerve cell. So this is about 2001 that we're now at, and I thought this technology would be ideal for the application that the physicians are asking me to try to fulfill, which is this multiple labeling, 10 to 12 colors, let's find these markers. But I had a pretty significant problem. The quantum dots that were the brightest, and we want lots of bright signal so we can see whether the patient has this marker or not, they're made in oil. And how many of you know what your body is made of? Water, that's right, it's not oil. This is a pretty big problem because I have to get this nanomaterial out of the oil and into the water in order to be able to use it for this application. So I was inspired by this paper I had read in 2002 where they had used a special coating on the particle, the quantum dot, called a micelle, and it allowed them to take these particles and inject them into a frog embryo and study how the frog developed. So I thought maybe we could do something similar. But instead of using lipids or fats like they did to make a micelle, we use something called polymer, and it's a block copolymer. So block copolymer means it has two parts. And one part of it really likes oil, and that's going to bind to the quantum dot. And the other part really likes water. And it's basically going to form a little cage around my quantum dot, allowing it to go into water. And this is very similar to how soap works. So you have oily fat on your plate, and you add the soap molecule, and it binds to the oil and lifts it off the plate and lets it enter the water. So same idea, okay? And our dream was, could we make a particle that had lots of quantum dots inside it so it would be really bright, which could then go into water so that we could use it on patient biopsy specimens. To do this, we use an approach where we make an emulsion. So what an emulsion is it's a mixture of oil and water. So we start out with our nanoparticles, our quantum dots, and our polymer in the oil phase, and we add to that water. And then to make our droplets small, we add a soap, or in this case, an emulsifier, and we shake it up to get really small droplets. Over time, the oil evaporates, and it leaves behind our particle that's surrounded by this micelle box. And so this is a video of this happening in real time. And if you look at the top right, you can see the oil droplet is slowly shrinking. And in a couple minutes, it's going to fission into multiple small droplets. There it goes. So that was kind of unexpected. And if we want to understand what's happening inside that droplet, we can blow it up. And so this is one oil droplet, and it's got polymer that likes the oil end and then the water-loving end. And it's got nanoparticles inside of it. And over time, those surfactant molecules make the droplet unstable, and it causes it to fission into daughter droplets. And this process repeats over and over again until we have really, really tiny nanoparticles. And why is that important? That's important because the parts of the cell that we want to label, these markers that tell you what type of cancer you have, are only about 10 nanometers in diameter, which is about 5 times the width of the DNA in your body. So we need particles that have a similar size to the marker so that we can detect them. And this is what they look like. So this is an image from a transmission electron microscope. And the white ring around the black dots is polymer. And then the black dots are the individual quantum dots that are inside of this micelle box. And this particle is about 25 to 50 nanometers, which is similar in size to our marker. So the next thing is we used particles like this to see if we could also image whole organisms, like we had seen in that 2002 paper. So we're now brought up to about 2010. And we used these particles to see if we could image inside fruit flies and zebrafish in the QStorm project. And I encourage you to go to the QStorm website, where you can find out more information about using these types of materials for imaging in whole organisms. But we're really interested in the cancer application. And so for this, we need different colors of particles, each of which would be bound to a different targeting agent that would bind the biomarker of interest. So in this example, we would like 3 markers, a red one that's going to look for normal cells, and then 2 cancer markers, green and blue, that are going to look for cancer markers on cancer cells. So I had been working on that project for about 3 months when something extremely unexpected occurred to me. I was in my bathroom, and I found a lump. So imagine my surprise. I went to the doctor, and I found out that I indeed had breast cancer at the age of 35. And it was a very interesting experience to be a researcher who understood cancer fairly deeply, but then become a patient. And it completely changed the way that I looked at research. During my treatment, I had the full treatment. I had a mastectomy of my right side with reconstruction. I had chemotherapy and radiation. And it's now been 5 years since my last treatment. So hopefully everything's okay. Knock on wood. Thank you. But what I learned from this process really completely changed the way that my laboratory did research. I'm an academic scientist, so the normal academic funding cycle is I come up with this really fantastic idea, and then I'm going to write a grant and try to convince someone that they should give me money to test this idea. And if I'm really lucky, I get money, and I go into the lab and we do experiments, and hopefully my idea works. And I write a paper about it, and I say, hey, I had this great idea, and it worked. And we all pat ourselves on the back, and we're ready to do the next big idea. So the main outcome here is publication. And that's it. Sitting there as a cancer patient, thinking about all of the technologies that I have in my own laboratory that could impact other people, and how we're making no effort to translate them to the clinic. So I decided we're going to switch focus and do something called translational science, which is a lot more difficult than what we were doing. It kind of starts out the same. I get an idea, I get funding, and I go do experiments. But we have a little bit of an intermediate step here. If I want to eventually impact patients, I'm going to need to commercialize my technology. So now I'm going to file for intellectual property protection, so I file for a patent. Then I can publish my paper. But now, instead of that being the end of the cycle, that's actually kind of the middle. The next step is to try to make enough material that I could actually test this in real people, which means that I'm going to need to apply for additional funding, and I'm going to need to scale up my process to make enough material to do a clinical trial. If I'm very fortunate, I'll actually have clinical impact and be able to translate my work. So the way that we were making these particles uses a process that involves probe sonication, which is basically a giant blender that we use to mix the oil and water phase. And when we did that, the product that we got made about 0.1 milligrams, and I would need 100 of those to do one patient. So this is not very feasible in the sense of something I would actually ever use in the clinic. And the next, okay, fine, I'm going to get a bigger blender, and I'm just going to try to make more, right? So we tried that, and we got that, which is not what we wanted. It was a white, murky mess. So I was stuck for a little bit, and then I remembered my colleague, Dr. Barbara Wuzlusel, who is also a chemical engineer at the Ohio State University, and I knew that in the past, Barb had published some papers where she was able to make particles similar to what I wanted using a process that involved a spray that was continuous, so it could make a lot of material. So I called Barb and I said, Barb, please help me.
Thanks, Jessica. As Jessica said, I'm also a chemical engineer, and I'm also interested in making particles that have some role or some aspect of biological research in them. So when Jessica came to me with this problem, we said, well, we've been doing a lot of work with electrospray, so let's see if we can use electrospray to solve this problem. So what we would do is we would take two little needles and stick one needle inside the other, and then we're going to put down the center, we're going to put that oil phase down the center, the oil plus the polymer plus the quantum dots. And then in the little space that's between the two needles, we're going to flow the water and the emulsifier. In electrospray, we add another force, and that is we just put a really high voltage between the needle and a ground ring. And what that does is it adds an extra force, so that big drop becomes a series of little drops, becomes a cone with a jet coming out, and that jet breaks up into a bazillion little drops. And then we collect whatever we made in that dish. So I'm going to show you a video of this process, and initially I want you to look in the middle, and you're going to see a brown oily droplet surrounded by clear fluid. And that brown oily droplet has the nanoparticles and the polymer in it. So there's our electrospray setup. You can see the inner droplet is the oil phase. We're slowly ramping up the voltage. You can see the big droplet becomes small droplets. The droplet at the end starts to deform. It kind of forms a cone. And then the droplets that are forming the spray are too small for you to see with this lighting. What we were hoping was that as that aerosol, that little droplet, flew through the air, maybe enough of the oil would vanish and we'd be actually able to make that micelle within the aerosol phase and only collect the micelle in the dish. Well, that didn't happen, but what we did do was we made a really nice emulsion. That emulsion then evaporated, and we ended up with the micelles collected in the water phase. What we did not end up was that white goop that Jessica showed earlier. We had a nice emulsion, and it's a continuous process, so that means it has a good chance of being able to be scaled up. As we move through doing the usual thing that scientists do, which is change the conditions, we found that we made spheres, which we were interested in, but we also made some strange shapes. And we wanted to understand from a fundamental view, when were we going to make strange shapes and when we were not going to make strange shapes. And if you want to understand something from a molecular view, it's really convenient if you can find yourself a molecular modeler. So we have another chemical engineering faculty member in our department, Dr. Lisa Hall, who's also here, and she's a molecular modeler, and not only that, she's interested in polymers. So we said, Lisa, can you help us? Can you model this kind of a polymer? One that has a water-loving end and one that has a water-fearing end, and can you stick that polymer into a water environment so we can see what's going to happen? Oh, and by the way, can you add some particles too? So she was kind enough to say, yes, we can do all of that. So here's the starting point. She's got her polymers suspended in an environment that the polymer is completely happy in. You can see the green end and you can see the pink end, right? So that polymer is happy. And since molecular modelers are actually masters of their own universe, they can say, all right now, we're going to change all of the solvent to water, and we're going to see what happens. So when we do that, what we see is we only see green. That's because only the green end of the polymer is out there happy with the water. Everything else is hidden away. And if you want to see what it looks like inside, you can just set the polymer to be transparent, and when you do that, you can find where those particles are hiding. So you can, from a molecular point of view, get a really good idea of what's going on in real life. At this point, we realized we had a pretty strong team. We had someone who could look at things from a molecular point of view. We had two scientists who were both doing laboratory work from very different points of view. And we also decided we should work with the Boston Museum of Science to help us understand both the sustainability of any process that we make, any of the environmental health and safety issues, and also to help us communicate our story to the public. So we were able to get funding from the National Science Foundation in this program involved with scalable nanomanufacturing, and we've been able to show both the process that I described earlier and several other alternatives are good ways to move forward and scale up the production of these materials that, again, are important for helping cancer patients. So where we are right now is trying to understand which of these processes works best and which one we should really move forward with. Now, while all this science was going on, Jessica was also interested in pushing forward the commercial side and making sure that this translational part of the science was also going forward. So I'm going to hand this back to her and let her tell you that part of the story.
Thank you. So now we're up to about 2012. And we decided we needed to found a company so that we could take our technology to the clinic. But the problem is, I'm an academic scientist. I don't know anything about running a business. I'm not an entrepreneur. So I was very fortunate that at this time, the National Science Foundation had introduced a new program called I-Core, or Innovation Core, specifically designed to train scientists how to speak business language and how to communicate with entrepreneurs and commercialize materials. So in this program, a student and a business mentor, we worked all together to basically talk to pathologists to figure out what they needed these materials to be able to do to be most effective with patients. We entered our business plan competition at our university, and we won the competition. So we had startup capital, and then we secured premises at a local technology innovation incubator in the Columbus, Ohio area. So at this point, we're now ready to ask the question, can we do what I showed you earlier? Can we label multiple markers in the same specimen using these quantum dot materials to identify cancer markers? So here are our results. At the top, you can see the combined image with all the different colors, and we can use computational algorithms to separate the colors into the 3 colors that were used in this image. So the red cells at the top right are normal cells that this patient has. And the cells that are green or white express one of the 2 cancer markers. So we were able to detect these 2 markers in these samples. So next, we looked at what kind of information could we provide a physician that builds upon what they already have. So this is just looking at one cancer marker, the white marker. And in addition to saying, yes, you have this marker, or no, you don't, we can provide quantitative information. We can provide actual numbers that correlate to how much different cells express this marker. And so this is the type of test that could be used throughout your therapy to see how effective a drug is. So you see on the left, we have our normal cells. And the goal is for that bar to be 100%. We want all normal cells. And right now, this patient has 5% of cells that are cancer cells. And so we would administer a drug, and then we would do this test again, and hopefully we see that bar go up to 100. But if not, it allows the clinician the opportunity to adjust your therapy. And that really is personalized medicine just for you. So I really would like to thank the team that's helped us with this journey. We are continuing down this process, and the company is hoping to make our FDA filing this summer. And Barb and Dr. Hall and one of the students involved in this research, Gori Nabar, are here to answer any questions that you might have. So thank you so much for your attention.